Hi, I'm Mark Yarchoan. I'm a physician scientist at Johns Hopkins and excited to talk to you about our work in fibrolamellar cancer.
Hi, everyone. My name is Marina Baretti. I'm a medical oncologist at Johns Hopkins, and I really specialize in patients with GI cancer, especially fibrolamellar cancer. And so it's really a pleasure to be here today.
What we'd like to talk to you about today is our work on a vaccine for fibrolamellar hepatocellular carcinoma. I think, as everybody knows, fibrolamellar cancer is driven by a unique fusion event between one gene called DNA JB1, the other gene PRKACA, and they come together. And I think that because all fibrolamellar cancers have the same genetic problem, it gives us a potential target to try to attack the fibrolamellar cancer cells. And there are different ways to do this, but our approach has been to help the immune system and educate the immune system to attack any cell in the body that has this particular event. I think what's unique about this is that every single patient with fibrolamellar cancer has this in their fibrolamellar cancer cells, but it's not in any of the normal cells in the body. And so that's an ideal target for the immune system because you can very specifically attack the cancer, but not attack normal cells. And so what our group has done is to make a vaccine that has a little piece of this abnormal protein, and we give that with two other drugs that turn on the immune system. One is called nivolumab, the other is called ipilimumab, and those drugs work to turn the immune system on while the vaccine points the immune system towards the cancer. And Marina, I don't know if you can discuss the results from our initial 12 patients that you presented at SITC this year.
Yeah, absolutely. I think, first and foremost, we need to state, and probably our audience knows, unfortunately, very well, that we don't have yet any standard of care or a very effective therapies. So really, this trial was born with the urgency to find a better therapeutic approach for our patients so that we can improve the quality and, of course, also the quantity of life. And so we did translate this very intriguing hypothesis coming from the lab into these very novel clinical trials, which, as Dr. Yarchoan just mentioned, was combining the peptide vaccine plus two checkpoint inhibitors that are already approved in other tumor types, including, liver cancers, which are nivolumab and ipilimumab. The design of the trial included two phases. The first phase was what we called the priming phase. The idea was really to set up the best priming immune response from the immune system standpoint. And then we had the maintenance phase. So during the priming phase, patients received this vaccine, which is in the form of subcutaneous injection weekly for the first month, and then every 3 weeks together with nivolumab and ipilimumab, which are infusions once every 3 weeks for a total of 4 times. And then patients went on to just receive nivolumab once a month and then a boost vaccination every 12 weeks. And patients could continue this combination of vaccine and nivolumab up to 1 year, of course, unless there were either not effectiveness or any toxicity. And of course, this was the very first time that we tried this vaccine and the combination with checkpoint inhibition. So our first question was to make sure that this combination was safe and well tolerable. And among the 16 patients that at least received at least one dose, first and foremost, we showed that this was absolutely safe. We really didn't see any unexpected toxicity, especially from the vaccine, which is really the novel drug here. The majority of patients just have some local reaction that totally resolved within a couple of days from the injection without any long-term sequelae. And then the second question was, are we actually providing any clinical benefit? And so I do want to mention that this, unfortunately, a majority of our patients came in when they already tried prior therapies, systemic chemotherapy, radiation. And despite all this attempt, the tumor was unfortunately growing and progressing. But then when they came into our trial, we saw that the majority, 75%, had some control of the disease. So the tumor was not growing anymore, was not traveling anywhere else anymore. And among these, we had actually 25% of patients that actually had the real shrinkage of the tumor, some of the lesions, for instance, patients with lung metastases or with lymph nodes totally even disappearing upon the treatment with the vaccine and the checkpoint inhibition. And actually, I do want to mention that even those patients that eventually came off study, the majority, they didn't come off study because they had worsening of the disease, but because actually there was such a nice control of shrinkage of the tumor that we were able to think about debulking surgery or some local therapy to try to be as aggressive as we could with the treatment. And then finally, third questions was, are we actually able to mount this good immune response that is specific towards the fusion? And in our trial, we had different samples collection. And first we saw that at baseline, so before receiving the vaccine, only one of the 16 patients was actually able to see the fusion in terms of immune response. But then after the vaccination, almost all the patients, 8 of the 12, actually did mount this specific T-cell vaccine, which we think is extremely important to even create that long-term memory to kill off the cancer cells, even in the future. One of the questions, and I think perhaps Mark, you can speak of, that we were a little puzzled, was that even some of the patients that had this response, then from a clinical standpoint, we really didn't see that nice tumor shrinkage that we would expect. And I think in your lab, you were able to find potential reasons on why there is this mismatch between immune response and then clinical response.
Thanks, Marina. I think you summarized our findings very well and the next step. So we know that there's a group of patients where the immune system saw the vaccine, reacted against the vaccine, and those T cells went to the tumor and the tumor shrunk. There's unfortunately a group, it's pretty small, where the immune system just didn't recognize the vaccine. And that's a group where we don't think that we achieved what we wanted to achieve. And it may be that some immune systems just don't see that little piece of protein that's on the cancer. But then there's actually another group where the immune system reacted against the vaccine. We see in the blood that there are lots of T cells circulating around. The T cells are the ones that attack the cancer most effectively, that are very specific against this piece of protein that's in the fibrolamellar cells. And yet the cancer didn't shrink. And so you have a cancer that has this marker, you have T cells that recognize that marker, and yet somehow the cancer didn't shrink. And why is that? And so really that is what our lab is working on now, is how do we get these patients who have T cells that should attack the cancer, how do we get them into the cancer more effectively and to attack the cancer? And what I think we've learned is that fibrolamellar hepatocellular carcinoma, it's a tough cancer in that it has certain ways of turning off the immune cells, the T cells that try to attack it. So even if you have T cells that float around that see the cancer, that doesn't always translate into tumor shrinkage. And what we're trying to do is figure out how do we make these T cells more effective within the tumor by turning off these negative signals that prevent the T cells from working. And so that's really the direction of our work moving forward. We think that one of the ways that cancer cells may do this is cancer cells secrete some things that are not good for T cells, including ammonia. So we know that fibrolamellar hepatocellular carcinoma often makes ammonia, and ammonia is something that is unfortunately pretty toxic for the immune cells within the tumor. Also, a lot of acidosis. So these tumors are high acid environments, and T cells just don't like high ammonia, high acid environments. The tumor cells also gobble up a lot of nutrients that the immune cells need within the tumor, and one of them is glutamine. And so, I think this presents a real challenge for the immune system, and this is what we'd like to do. We'd like to keep building on what we've done. We know that there's some patients that appear to be benefiting, but we want to increase the percent of patients who respond to this treatment, hopefully by adding something else in. And so that's what our work is focused on moving forward. I'll just talk very quickly about what's next for our vaccine program for fibrolamellar HCC. We are very encouraged by the activity that we're seeing. Our eventual goal is to make this as accessible as possible for patients. And really, eventually, we're going to need to find a pharmaceutical partner who can really make this available outside of Johns Hopkins, where we are. In the meantime, with support from the NIH and also from the Fibrolamellar Cancer Foundation, we've been able to expand our trial. And in this expansion, we've actually been able to loosen the criteria a little bit for who we're allowed to treat. So when we started the trial, because all of our mouse work that supported the trial was in mice who had never received a drug like nivolumab before, the trial in the past was restricted to patients who had never seen nivolumab. But actually now, with the results that we have, we've been able to go back to the FDA, and the FDA has allowed us to loosen the criteria to allow patients who've received checkpoint inhibitors in the past. So we now have the trial open for anyone, including patients who've had nivolumab in the past, as long as their organ function is okay and as long as they have a tumor that we can actually follow. The other thing, and this is a little less relevant for patients with fibrolamellar cancer, but the same fusion event that drives fibrolamellar cancers also can be found in some other rare cancers, like some cholangiocarcinomas and pancreas cancers. And so we've actually been able to expand to allow other patients who have a fibrolamellar-like cancer with the same fusion to participate in our trial. And the very last thing I'll just say very quickly is that as we treat more and more patients, we're finding T cells within our patient's blood that now have been trained by the vaccine to attack fibrolamellar cancer. And what we hope to do is to take those T cells from our patients and actually turn those T cells into the next drug. And so the patients who participate in our trial, their T cells will be able to clone those T cells and actually put those T cells or those clones of those T cells into other patients with fibrolamellar cancer. And so, I always say this to patients, a trial by definition is something that is new and we don't exactly know whether it's going to work. But the one thing that we can guarantee our patients is that we're learning from every single patient and that they really do have the ability to help future patients by becoming a drug themselves, which is a pretty wild thought. So hopefully a lot more progress for our patients. We're really excited by the progress we're making, and thank you for the support.